All right, ladies and gentlemen, welcome back. So we're talking about cells, and if we keep in mind the scale we're talking about here, cells are microscopic, hence the reason we need things like microscopes in order to see them. And there's a reason for why those cells need to be that small. And it really comes down to the ability for cells to move things across their cell membrane. All right. And everything that goes into a cell, everything that goes out of a cell has to be able to cross that membrane, what is essentially its surface area. So, in order for cells to be efficient, right, they need to get nutrients like oxygen and sugars and proteins and things like that into the cells so that they can do their job and function. They also have to eliminate waste products like carbon dioxide, for example. All of that stuff has to cross that surface area. The smaller that cell is, the greater that surface area is compared to its volume. So the bigger that cell gets, as that volume increases, its surface area doesn't increase at the same rate. Okay, so the volume is going to be able to increase at a faster rate than that surface area does. So the bigger the cell gets, the more volume it has to the same surface area. That's going to decrease its efficiency. So when the cells get to a point where they are not efficiently enough moving materials across that surface area across its cell membrane, that's when they start making more of themselves. And that ratio of efficiency to that volume and surface area certainly varies depending on the cell and the cell's function. But generally speaking, our cells are small because we need to move things across it and we need to do it effectively. When we get to talking about cell transport, we'll come back to this idea a little bit more because we're going to talk about how cells transport things across that membrane. And that's really where that efficiency comes in. To be able to do that, they need to be able to maximize that surface area to volume ratio in order to be efficient. So let's start talking about cell classification here. All right. And so when we look at cells, they really come in a handful of categories. And the first thing we look at is it a prokaryotic cell or is it eukaryotic? And if you remember talking all the way back in the first unit, what's the difference? Well, prokaryotes have a nucleus. Excuse me, they lack a nucleus. Prokaryotes lack a nucleus. Eukaryotes have a nucleus, right? So these guys have no nucleus and really don't have a whole lot of internal structure either. So we're looking at something like these guys here. Your eukaryotes, on the other hand, do have a nucleus, as well as a bunch of other internal structure. So things like this guy here and this guy here. All right. And that lack of internal membrane or having it has a huge impact on how big those cells are. So go back to that surface area to volume ratio we were just talking about. Eukaryotic cells are capable of being bigger simply because they've got that extra internal membrane to increase their surface area and as a result increase their efficiency internal to their cell. Your prokaryotes are going to get limited much more heavily by that surface area to volume ratio because they don't have any of that internal membrane structure. Your eukaryotic cells then get classified even further. Are they plant versus animal? And that comes down to a subset of materials, subset of organelles that they contain. So plant cells have things like your chloroplasts, right, that we see right here. Central vacuole, for example. So this guy is a plant cell over here, right, and this guy over here lacks a lot of that extra components, but has some of its own, and those are the animal cells. So if we take a look at our prokaryotic cell, again, there's not a whole lot of internal structure going on here. They have some basic components. Your nucleoid region sits there in the center, and that's going to be the housing for your genetic information, all right, your DNA. They do have ribosomes. We're going to see those ribosomes show up in eukaryotic cells as well. And those ribosomes are going to be really important for your cells to be able to use that genetic information in order to make proteins. So remember, we were talking about that whole idea of DNA to RNA to amino acids, right? Well, that's what your ribosomes are there for. All right, they're there to take that genetic information and then use it to build the proteins. So that's where our protein synthesis comes in. Your cytoplasm, also going to be something we see in your eukaryotic cells as well, just kind of the place to hold all of this stuff. It's kind of that internal space. It does sort of have a jelly-like consistency, mostly water and other dissolved molecules. They do have a cell or a plasma membrane that'll always be the most internal covering. It's the layer next to your cytoplasm for all intents and purposes. Prokaryotic cells also have a cell wall, which will be right outside of that. Cell membrane is going to be really what drives what can go in and what can go out. We'll talk when we get to cell transport about the idea of your cell membrane being semi-permeable. It lets some things come in and keeps others out. Your cell wall sits outside of that, and that cell wall is really just there to be some extra layers of protection. Prokaryotic cells, a lot of them have a unique structure, one layer outside of that cell wall, and that's this thing called a capsule. And that capsule usually is a bunch of carbohydrate molecules, sugar molecules, and it is literally like being stuck in a sugary, gooey substance. And so they use it to stick to things. It's how they adhere to different surfaces, whether they're hard surfaces like you would think of like furniture, doorknobs, that kind of thing, um, but also to surfaces inside maybe organisms that they live in or more moist environments in just the general ecosystem. Some prokaryotes can also have these pili, which are these little hair-like projections sticking off that cell wall capsule area. Those pili are interesting because they are used for adhering to surfaces as well. So similar to that idea of a capsule, they can kind of grab on, kind of think of them like little spaghetti bits with suction cups on the end. And they can suction cup themselves to spaces so they don't get washed away. But they can also use those pili in certain situations. Certain types of bacteria will actually use them to connect themselves to another bacterial cell. And so they kind of almost end up acting like a straw shoved into two cells. And what they do is they'll share genetic information back and forth through that pili. And when it's used like that, they talk about it as a sex pilus, and it is the closest thing that bacteria have to sexual reproduction. Um, Prokaryotes are technically asexual, meaning that they are themselves capable of making their own offspring. But in that sense, go back to evolution. If I make a copy of myself, which is essentially what asexual reproduction is, that means I'm genetically the same as my parent. That also means that any siblings that I would have would be genetically identical as well. While that does present a little bit of a challenge when we get to talking about needing that variation in order for natural selection to work. So prokaryotes have found some interesting ways to get around the idea that all of their parents and all of their offspring, the entire population, should be genetically identical to each other because they're asexually reproducing. And one of those is to produce these sex pili and share genetic information not only between individuals of the same species, but the genetic structure of prokaryotes is way more flexible than it is for, say, us. And so they can actually share genetic information across species too, which is pretty cool. We'll hit on a couple of those pieces here and there throughout the school year as it comes up. But bacteria are pretty cool things when you really start studying them. The other thing that these guys can have are prokaryotic flagella. If you take a look at the picture, you see a handful of them coming off of just one region. These guys are literally utilized for motion. This is how these guys move. Again, this picture is just a model, so these cells don't necessarily look like this picture. but it gives you the basic idea. Some bacterial cells, some prokaryotes have all of these structures, some of them only have some of them. Those flagella, for example, can actually come in a lot of different orientations and varieties in terms of not only the number that they have, but where they get stuck out of the cell too. So lots of interesting stuff when it comes to looking at bacteria, what they do and what they look like. Okay, so let's just talk basics here because that's really what the point of this section is just broad overviews. What's potentially in a cell, what does it do, and what are the basic differences? So if we just stick here to comparing prokaryotic versus eukaryotic cells, right? We look at those prokaryotic cells because they're lacking all of that internal structure. They're going to be relatively small. They're also relatively primitive. Remember, going back to evolution, we believe these were the first type of true living cells. Eukaryotic cells came later. So they are smaller. They're probably older evolutionarily as well compared to those larger and more advanced eukaryotic cells that have all of that internal organization. They have those membrane-bound organelles. They've got those specialized cell parts that your prokaryotes just don't have. And they probably got them basically by eating them, right? Remember your endosymbiotic theory of the idea of how we got things like mitochondria and chloroplasts, right? One prokaryote ate another, and they just sort of had this mutualistic relationship as a result of that smaller prokaryote never getting digested. So when we look at prokaryotic cells, right, we're talking things like bacteria, as well as that domain Archaea So all your prokaryotes fall into those two domains. Your eukaryotic cells fall into that eukarya domain, and then we start talking, flushing them out by kingdom, so plants, animals, fungi, that kind of thing. The last thing I want to talk about here is the difference between their genetic structure and the way that their DNA is organized. So when we look at your prokaryotic cells, it's circular and is centralized in that nucleoid region there in the center, right? So remember, this is that nucleoid region right here. So there is no membrane that surrounds their genetic information and keeps it separate from everything else. Where when we look at your eukaryotic cells, right, they have a true membrane-bound nucleus. And so their DNA that sits in there does look a little bit different depending on whether we're talking prokaryote or eukaryote. So let's dive into that just a little bit more, and you should find some space on your notes to add this information, okay? So let's talk just real briefly about the differences between the structure of prokaryotic DNA all right, versus your eukaryotic DNA. So as we get into things later in the school year, we'll focus mostly on that eukaryotic stuff. So I wanted to take a few minutes just to talk here. First and foremost, let's just talk about the amount of genetic information. So we'll talk about chromosomes, right? The number of these stretches of DNA molecules that exist in a cell. When I look at a prokaryotic cell, they only ever contain a single chromosome. Okay, and it doesn't matter what species we're talking about. When I look at eukaryotic DNA, on the other hand, they're going to contain at least two or more chromosomes. And most eukaryotic cells contain considerably more than that. If we talk about humans, humans actually have 46 chromosomes. Right? You go to a different species and that number will be a little bit different depending on what you're talking about. And then there's also the structure of it itself. So I mentioned to you just a second ago on the previous slide that prokaryotic DNA is circular. What that really means is you take that double helix of the molecule and it literally bends back on itself. So in a really rough drawing here, okay, here's my molecule and it's going to bend back on itself, and it's that double helix, so you'll still get that twisting molecule that you would expect. All right, but it bends on itself and so that it links back together on the other side. Okay, so it's got circular DNA. Okay. When we look at the genetic information for a eukaryote, it's considerably more complex. All right, first and foremost, they have linear DNA. Okay, so that means that that double helix, right, exists in single lines. Okay, so they have linear DNA. When that cell isn't dividing, right, we're using that genetic information is all spread out kind of like you see it here. All right, so when we talk about that spread out DNA, all right, and we're accessing it for information, we'll talk about this as chromatin. All right, and basically this just means the cell is using that DNA. All right, so it's accessible. Um, when we talk about moving that genetic information, right, and we really use that term truly as a chromosome, all right, you often end up seeing it more like this. Okay, and these are basically the same genetic information that's duplicated and it ends up in these X structures. And this is what we would call a condensed form of that chromatin, what you would call a chromosome. All right, and we'll condense this genetic information so that we can move it, or we just don't need to access it. So we're in this form when we're moving DNA, or we don't need access to the information present. Okay, so how do we do this, right? Well, think of eukaryotic DNA like a big slinky, right? So when I have that slinky all stretched out, that's what I'm talking about here with this chromatin, right? I'm stretching it out, I'm playing with it, I'm using it, all right? But when it comes time to wanting to move that genetic information, I don't want to take that slinky all stretched out and go down the middle of the hallway at school with that slinky all stretched out between classes, right? It's going to get broken. It's going to get tangled. It's going to run into people. And you're never making it from one end of the hallway to the other stretched out completely without damaging the slinky. Well, same idea for your DNA and your cells. We don't want to do that. And so your cells will condense it, basically push it all together, right? Condense the slinky back into its smallest form. And that's what we're talking about with those chromosomes. So that we can move that genetic information without damaging it. We also do the same thing when we don't need access to a certain component of the DNA. All right. So we have along the entire stretch of DNA these little protein balls called histones. All right, so this is a histone. And those histones are kind of like magnets. Your prokaryotic cells don't have these histones. And so this is something that is unique for the eukaryotic DNA. And think of them as magnets that you can turn on and off. This is the way your cells are able to condense information, condense that genetic information, or stretch it back out. If I magnetize, quote unquote, those histones, I make them sticky and they stick together, that's when I condense. If I relax those histones and make them unmagnetized, then everything kind of just spreads back out. And this is part of how your cells are capable of differentiating. So when we get to multicellular organisms, right? If I look at your brain cell versus your skin cell versus your muscle cells, all of those cells are going to look and function a little bit differently. And that has to do with the information that they're accessing, not the information that's actually present. So you start off as a single cell, right? Egg plus sperm gets together those 46 chromosomes, and then that cell is going to go off and divide and eventually make the baby, and you continue making cells as you get older so that you can grow and develop. That means every cell in your body has all 46 chromosomes worth of information, but I don't need to access it all. So as your cells start differentiating and becoming brain cells, becoming muscle cells, becoming skin cells, they shut off the pieces of that 46 chromosomes that they don't need. So your brain cells are only accessing the components that make them brain cells. The rest of the information to be any other cell in your body is condensed and not accessed. So in your brain cell information, how to be a brain cell, that piece is open like this. Everything else is condensed. I go to your muscle cells and it's a different component of your DNA that's being accessed. Brain cell function and everything else is shut down. And hopefully that makes sense. And that's how we are able to specialize your cells. And we do this by telling your genetic information which set of histones to turn on, magnetize, and condense because I don't want access to that information because it's not relevant to my job. Or I unmagnetize those histones and let the genetic information spread out so that I can access it because it is relevant to what I do. So it's pretty cool, pretty unique, pretty elegant system for the way that your cells have taken care of this problem. Okay, so the rest of what we're going to talk about are the eukaryotic cells themselves and what is in those cells, what's not in those cells, what makes a plant cell different from an animal cell, and just the basic functions of the organelles themselves. So when we look at eukaryotic cells, right, we're going to split them out from plant, all right, versus animal. So plant cells here, animal cells here. And keep in mind, again, we're looking at models, just like we talked about with those prokaryotic cells. These are models of cells. They are not exactly what a cell would look like. These are going to look different depending on the specific plant cell or specific animal cell that you go after. The models are just there to kind of show us everything so that we can get a sense of how to study them, right, and how to teach them. So when we look at your plant cells, your eukaryotic cells, when we look at your eukaryotic cells, there are some components that are consistent across the board. And truly, these are consistent in some cases from every plant cell, every animal cell. They're all going to contain at least these components in general. First one is just your cytoplasm. This is that jelly-like substance that's inside your cells. It contains the molecules, the chemicals, your organelles. It's just kind of everything inside the cell membrane. If we specifically talk about the fluid portion of that cytoplasm, then you may come across the term cytosol, and most of that is just water, and then the dissolved solutes, the sugars, the proteins, ions, salts, that kind of thing that are dissolved in it. The organelles are those membrane-bound structures. These are the specialized components of the internals of your cell that have very distinct processes, that you'll have more of certain organelles in one cell type and less in another, depending on the function of that cell as well. And that's what we're going to spend a huge component of the rest of these notes talking about, are what are those organelles and what do they do. Last piece is also having a cytoskeleton. So think about what that term means. You probably know what a skeleton is, right? Bones. What do those do for your body? They give your structure, support, allow you to move, right? And so your cytoskeleton is just that. It's a skeletal system internal to your cell. And so it's a network of proteins that help support the cell, give it shape, anchor things in place, be able to let it move,